I was worried the last day there'd be no one here, but that is apparently not the case. So again, I just want to thank the organizers for the opportunity to present some of our work. Today I'll be presenting our shared structural variant mutagenesis mechanisms in cancer and the constitutional genome. And I have nothing to disclose. So from a very general sense, structural variation in the genome can be conferred as a copy number loss, that's a loss of genomic material, a copy number gain, where we copy just a piece of genomic material, or an inverted genomic segment. Again, I always say this is the slide I show my parents because it's a very simple view and is, I think, easy to understand structural variation from a simple sense. Unfortunately, there are more complex patterns of genomic rearrangements that can occur, and these occur because of inherent genomic instability points that are present within the genome. And again, this is the slide I show when I tell my parents why it took 6 years to finish a PhD. And again, we study it in the constitutional genome, but there are structural variation that's present in cancer genomes that's now being well studied by other groups to date. And so in our study of constitutional disease, we study really 2 disease cohorts where CGRs are very present, and that's MECP2 duplication syndrome and Potocki-Lupski syndrome. So I'm going to start with our work on MECP2 duplication syndrome. So MECP2 duplication syndrome is an X-linked genomic disorder caused by copy number gains encompassing the gene MECP2 on the X chromosome. It's 100% penetrant in males, and females can be carriers that are asymptomatic. And kind of the main phenotypic trait is very profound developmental delay and intellectual disability. And so as you can see from this chart, and this is a biobank from the Carvalho and Lupski laboratories, there's a wide range of structural variation that can occur at this locus, including just simple duplications, translocations, terminal duplications. But what I want to focus on today, and what we've really focused a lot of our work on, is looking at a very complex structure known as the dupe-trip inverted dupe structure. And so this structure was really beautifully shown by Claudia Carvalho in a 2011 Nature Genetics paper, where she showed the copy number state of the triplication was actually inverted within this segment that formed 2 breakpoint junctions within the structure. And so just if you're not used to looking at array CGH data, right, we're going from a copy number neutral up to a duplication, up to a triplication, back down to a dupe, and then back down to normal. And this structure, even though we have 4 points of copy number change, forms just 2 breakpoint junctions. And so I kind of wanted to show here is that downstream of the gene MECP2, which sits right here, are these inverted low copy repeats highlighted by the yellow arrows. And these actually act as recombinant substrates that recurrently form this complex structure. And so I wanted to just kind of make this cartoon. Again, we're on the X chromosome in a male, so we have our 2 arms. And the inverted repeats are shown here by K1 and K2. And these repeats at this point in the genome are about 11 kb in size and 99.9% similar and sit in an inverted orientation. And so you may wonder, how does this happen once, let alone recurrently? And so we try to draw kind of a schematic here. When we go through replication, right, and we hit this LCR, there's basically a stalling of the replication fork, and it jumps to a point of homology on the other, so here K2, on the other LCR. We then form this chimeric LCR, and the replication goes backwards before we hit another break highlighted by here. The template jumps back to the correct stand, and we finish replication, forming the dupe-trip inverted dupe structure. And so a little bit of a problem when looking at this structure, right, is that we have basically big gaps of repetitive regions that form the structure. And so during points of copy number uniqueness, right, we can form a breakpoint junction very easily, but when we hit those low copy repeats in other regions within this loci, we basically have no information as to what's happening by short read sequencing or by array CGH. Furthermore, right, we don't actually know which part of the segments are inverted in addition to that triplicated sequence. And so what we see here, right, is that we have breakpoint junctions that are identical through these 4 options, but we can't properly phase which genomic fragments go within the structure itself. And so we really, for this cohort, wanted to say, can we better characterize the mechanism of formation that forms those junctions within repetitive sequence, as well as identify the SV haplotype substructures within this cohort. So we really threw everything that we could at it, including high-resolution array CGH, short-read whole genome sequencing, optical mapping that was very beautifully shown in the previous presentation, Oxford Nanopore, and PacBio Hi-Fi. And so luckily I don't have time to explain optical mapping, but it was very nicely explained again previously. And so what we wanted to see here, I just wanted to take one sample to show how we do this type of analysis. We can see up top, I put the array CGH for this sample with a dupe, trip, dupe, right? An array gives us very nice information for the copy number, but it doesn't give us any architectural information of how that actually is placed on a linearized chromosome. And so what we can then start to do is build our data from our array and looking at our optical mapping data. And I put here just kind of the arrows where those copy number states are. And what we can start to see is that there are inverted segments connecting within the structure itself. And what's really, really nice is that we can actually see both breakpoint junctions in a single DNA molecule. And so down here in the optical mapping data, we basically have single molecule resolution. And when we study structural variation, we really want to study it in cis. And so it's really, really nice to have single molecules that show the entire structure on a linearized chromosome. And so when we do that for this specific sample, we can see that it's not just the triplication that's in an inverted status, but it's actually the triplication as well as that initial duplication, including MECP2, that are both inverted. So if we go back to our possible substructures, right, we can actually see it matches very nicely this second substructure right here. And when we go through the rest of our samples, we can actually find all the substructures that are present within our cohort. And very interestingly for us, right, MECP2 is actually in an inverted orientation in several samples. And so that's the extra copy of MECP2 is inverted. And so right now we're trying to see, is there a phenotypic correlation with MECP2 inverted versus in direct orientation, and to see how transcriptionally active that copy of MECP2 may be. And so that's great for the SV haplotype structures, but we also wanted to see, can we have nucleotide level resolution through these repetitive regions? And that's really where we employed nanopore and hi-fi data to help us view. And so what we can see here very nicely in unique sequence, we can see these soft clip reads. And so that's basically a read where one side aligns to reference, the other side aligns to the other side of the junction. And so within unique sequence, we can see it in short read as well as everyone else. But when we move over to the point of copy number change that's in non-unique sequence, so this one sits in a gene repeat cluster, short read basically gives us nothing, right? The mapping quality is 0 because it's too repetitive. But our nanopore and hi-fi sequencing data very clearly shows us where the breakpoint junction is and allows us to form a breakpoint junction sequence for this sample. And so what really we wanted to see is, can we have a visualization through those LCRs, right? So they're about 11 kb, and so our hope was that we would be able to have reads that spanned the entire LCR, that were anchored in one side, and then gave us a view of the other side. And so I just wanted to kind of show that region here. Again, when we look at our short read whole genome sequencing, we have no mapping quality, right? Because it's a highly repetitive sequence that we're not sure where to map these short reads. When we look in nanopore and hi-fi, though, we can see that we do have reads that span the LCR, starting in one side, going through, and ending in the other. And then what we can start to do is go through those reads and say, do we see reads that have soft clipping? And so what happens is that chimeric LCR, the breakpoint is happening somewhere within this LCR, but since it's 99.9% similar, we don't see it. But what we do see is that when unique sequence gets out of the LCR, we then have soft clip reads that are the other side of the junction. And so what we're seeing here, right, is this read represents this chimeric LCR that's being formed. So the reason that we're able to do it with hi-fi and nanopore is because it's actually anchored in unique sequence before the LCR, moves through the LCR, and then we have non-unique sequence moving out. And it allows us to actually predict which LCR was used to generate the actual dupe trip event. And so where we really started to come in and say, wow, this is a very interesting pattern, but then when we start to look at the cancer work and cancer genomes, we can actually see this dupe trip is one of the 12 most prevalent mutational signatures also in human cancers. And so what we're starting to explore is that these inverted LCRs, which are very prevalent within the genome, not just at the MECP2 locus, actually act as points of genomic instability in human disease, such as MECP2 and others. But what we're starting to see is it's also prevalent in cancer genomes and can act as points of genomic instability. And so very quickly, I just want to go through one more cohort, and this is the Potocki-Lupski syndrome cohort. So Potocki-Lupski syndrome is caused by copy number gains involving the gene RY1. The main phenotypic feature is developmental delay, as well as failure to thrive, and some other neurological issues. And so as you can see here, like our MECP2 duplication cohort, we have a very diverse set of structural variants that can occur within this cohort. But what I really wanted to focus on, which is a really interesting part of our study right now, is copy number gains that are near that gene RY1, but don't actually cover the gene RY1, that were diagnosed with Potocki-Lupski syndrome, but our question was, do they actually have Potocki-Lupski syndrome? And so what I wanted to show here is just kind of the architecture of this region of the chromosome, as well as the samples that we've been starting to collect. The gene RY1 is right here, and you can see we have many samples with copy number gains and a broad phenotypic spectrum of Potocki-Lupski syndrome, but don't actually have a copy number gain over the gene itself. And so what was really interesting to us is that we actually have a collection of samples with identically overlapping, we actually have 2 more samples coming in as well, with identically overlapping copy number gains and our higher order amplifications, right? So this is representing a 6x amplification as well as a triplication and a duplication over the exact same genomic sequence. And so I just kind of wanted to show here an array CGH view showing the region. So this is the Potocki-Lupski syndrome common duplication that's mediated by repeats flanking both left and right. But what happens is that those overlapping samples actually have a breakpoint that occurs within this sequence right here. And so what we started to look at is basically where that breakpoint is located and what might be there. And what we found is that it's actually at the ISO17Q breakpoint cluster. And so this breakpoint cluster is very interesting because it's actually a polymorphic region with repeats in an inverted and direct orientation. And it's actually the most common abnormality within human neoplasias. And the mechanism is basically leading to non-allelic homologous recombination in cancer that can lead to these isochromosome 17q formations. And so as it's been studied by Claudia in the laboratory, this is a highly polymorphic region with, again, direct and inverted repeats that mediate that cancer formation. But for us, we see this as these repeats, especially these direct and inverted repeats, as mediating the formation of these higher order amplifications within this sample. And again, when we actually look at these samples, it's interesting because we have an oscillating copy number state at the end, suggesting there's a rolling circle type amplification to form these higher order events. And I wanted to show this slide again because I think it really represents that this local genomic architecture is causing genomic instability, not only in the constitutional diseases that we study, but is also leading to instability within cancer genomes and leading to structural variant formation within those samples. And just some quick conclusions. Inherent structural features can lead to instability and SV formation. And again, what I really wanted to highlight was those repetitive regions, those inverted LCRs, especially at the MECP2 locus and others. In constitutional disease, can lead to this structural variant formation, but also in cancer, the instability can lead to structural variant formation using those same recombinant substrates. The patterns dictating SV formation are shared processes, so the dupe trip inverted dupe is found not only in constitutional disease, but is also one of the 12 most prevalent mutational signatures in cancer. And all of this kind of drives our understanding of how these mechanisms work in all disease states, cancer as well as constitutional disease. And with that, I just want to thank my mentors, especially Claudia Carvalho and James Lupski. It's really an honor and privilege to have been mentored by both of these phenomenal scientists and everyone at Baylor and elsewhere who I have worked with on these projects. So thank you so much, and I can take any questions.
Okay, the floor is open. Yeah, go ahead.
Hey Chris, Alex Heuschen from Nijmegen. That was outstanding, fantastic. Have you looked at the telomere to telomere? Were they in very tricky regions that may be even able to help you a little bit further? Is it more accurate also for LCRs?
Yeah, so we actually just got the telomere to telomere reference in our optical mapping data. So we're working on realigning those samples, but I think that is kind of a low-hanging fruit that we want to see, like, does it help us in some of these like complex regions? So ongoing,
Right.
Yeah.
I'm certainly interested to see what you get when you resolve the phenotypic differences of the different substructures around MECP2. That's certainly an open question.
Yeah.
I was glad that you immediately answered that.
Yeah, because I think it's something where there is a phenotypic spectrum, and it's not related to duplication size. And we know that. So we are saying, are there other things that we can kind of think of that are stratifying these samples in different categories that we can kind of work upon?
Excellent, exactly. So with that, thank you very much. We're going to move on.
So, the next speaker is